Working? All right. So thank you, Raj. Thank you very much, Raj Umesh, for organizing this meeting and for this invitation. This is a privileged opportunity to visit a place of the world that I'm not sure I would have gotten a chance to see. So I really do appreciate that. I'm going to go through some background information about stress granules and neurodegeneration and why they have a particular focus in the neurodegenerative diseases called ALS, or amyotrophic lateral sclerosis, and frontotemporal dementia, two diseases which we now understand to be linked pathogenically. So my background is in biochemistry. I'm not a neuroscientist, as you heard. My background was studying how cells die. And so every cell has a programmed pathway for death, one or two or three different pathways at this point. And so in the context of cancer, cells don't die when they should. And in the context of a disease like ALS or FTD, we have neurons or cells, specific types of cells that are dying when they shouldn't. And all of those cells, they experience stress. And so every cell is constantly being bombarded with stress, and the cell has to make a decision all the time. Should it live or should it die? There's various stress stimuli that can come both from intrinsic and extrinsic factors. And so when we think about extrinsic factors, we're talking about things that are in the environment, for example, like temperature fluctuations, extreme heat, extreme cold. And so our cells are wired to have very particular responses to these things. Pathogens, if we think about, we're constantly being bombarded with viruses and bacteria. Fortunately, most of them we don't actually experience any symptoms with, and they're subclinical, but it doesn't mean that our cells are not working hard to make sure that they limit the impact of those pathogens. And of course, in the environment at large, we're constantly being exposed to chemicals, whether it's in our food, in our water bottles, in the clothes that we wear, and lotions and all that. Our skins are constantly being bombarded with various types of chemicals. Some of these are generating reactive oxygen species. Some of them might contain heavy metals. And those are just a few. We can also count UV rays from the sun, or also another example of extrinsic stress. Intrinsic stress, most often in the space of neurodegeneration, we think about misfolded proteins. And so there are many examples of neurodegenerative diseases that feature particular proteins that they just suddenly, they do not have the normal conformation. And so they're not functional. That puts stress on the cell in order to try to quickly refold them and make them functional again. And so if you think about it in the context of Parkinson's disease, this might be alpha-synuclein. In the case of Alzheimer's, this might be A-beta and APP. And in ALS, there's a number of different possibilities, including superoxide dismutase 1, TDP43, and many others. So every time the cell encounters these stress, there has to be a decision made whether or not the cell is going to live or whether it's going to die. So to be or not to be. So the consequence of this decision is going to be very different for different types of tissues. And so if you think of a cycling cell versus a terminally differentiated cell type, the consequence is going to be different. So in our skin cells, for example, which are constantly cycling and constantly replacing the outer layer and just constantly being exposed to UV irradiation, for example, it's okay because we have a regenerating pool all the time. But if you're thinking about neurons or cardiomyocytes, so the cells that, the muscles that drive the heart muscle, these are terminally differentiated cell types, which means they have one life to live. And once that decision is made that they are not going to survive the stress, that's it. They're not getting replaced. And so many of the neurodegenerative diseases that we think about is actually selective loss of particular pools of these types of neurons, of different types of neurons. And the question is, why some neurons are lost in one disease versus another? That's the holy grail. We don't actually really understand that as a neurodegeneration field at large. So that's an important consideration when we talk about how cells are going to manage stress. So cells are equipped with many different ways in which to overcome these various extrinsic and extrinsic stress exposures. One is a pathway is referred to as the integrated stress response, and that's because it integrates multiple different pathways. So, there's lots of different ways into the pathway. And so depending on what the trigger might be, and here we've listed a few, so amino acid deprivation, so arsenite, which was a type of oxidative stress, osmotic stress, viral infection and heat shock, or this, what I talked about was the protein misfolding, they're going to activate particular types of kinases that eventually are going to have a function, they're going to function on EIF2 alpha and cause phosphorylation. This phosphorylation event is essentially going to trigger the disassembly of polyribosomes, so that translating RNAs that were on the ribosome plus ribosomal components, they get kicked off and they end up getting accumulating in these macroscopic or microscopic granules called stress granules. The other consequence is actually you're going to get activation of ATF4, which is a transcription factor that's going to drive the expression of a number of cell survival genes. So in general, this pathway is considered to favor adaptation to various conditions. And you see that stress granules are very much integrated into this pathway. Now, there are examples where this pathway actually does not favor adaptation and, in fact, results in the activation of apoptotic signaling, but we're not going to cover that here today. So what are the stress granule pieces? So in this video that I've borrowed from a published paper, is cells that have been exposed to sodium arsenite, and they're expressing a tagged version of a protein called G3BP1. And so you'll see that initially that the signal is very, very homogeneous and distributed throughout the cytoplasm. And over time, there is a coalescence of the signal into these cytoplasmic puncta. They start out first very small and then later grow to larger entities. These structures are very conserved. They were actually first identified in plants, I believe somewhere in the 80s. As the video shows you, they are transient. In fact, when you remove the stress, these structures are going to basically disassemble. It's considered that these structures form through liquid-liquid phase separation, which is this concept of changes in viscosity and cytoplasmic subcompartments. And so if you think about when you make a vinaigrette, say oil and vinegar, and you have the oil droplets that are going to separate out. So it does not require energy for these structures to form, and actually can be driven by increasing the cytoplasmic pool of non-translinked mRNAs. And so as I mentioned, how they get kicked off of the ribosome following the IF2 alpha phosphorylation. The composition of these granules is vast. In fact, there has been endless number of efforts that have tried to catalog all of the components that are in these stress granules. There are lots of different polyadenylated mRNAs as well as long non-coding RNAs, tons of different RNA binding proteins and pre-initiation translation complexes, as well as proteins that are thought to be more relevant to signaling is another subset that are included. One important thing to remember is that the composition of these stress granules actually will vary according to cell type, as well as according to what the stress signal is. There is a common core of stress granule proteins, but in general, you will find thousands of different types of proteins in the different components. And as I mentioned, that these are rapidly disassembled once the stress is no longer present. And actually, this is an ATP-dependent process that requires either chaperones or ATPase-driven chaperone activity, or some people suggest that there's also autophagic engulfment of these granules to disassemble the components. So what do they do? This is actually the million-dollar question in the stress granule field, because there's no real demonstration of what they do. The classical view is that they are storage sites and or triage sites for these polyadenylated mRNAs whose translation was suppressed during the stress exposure. This has caveats, though, because it has actually been demonstrated that some RNAs can actually be translated within the stress granule, and that was demonstrated using some very nice single-cell imaging. There's also been proposed that, speaking to the triage component of this view, is that maybe that these structures actually serve to sort RNAs and transfer them to other RNA granule compartments. This hypothesis has been around for probably 15, 20 years, and actually there is no experimental evidence to support that actually that is what's happening. There is demonstration that these stress granules can seem to interface with other granule types, specifically processing bodies, but the actual demonstration of an individual RNA transferring from one granule to another has never been shown. In general, as suggested from the initial work in plants, it suggested that these are a pro-survival mechanism, and that this might be due to sequestration of apoptotic factors. It might be because there is ribosomal priority, translational priority given to pro-survival RNAs that are to be translated. And it's also been shown to be an inhibitor of viral replication, although there are many examples in which viruses actually hijack these components for their own advantage. So that's the classical view. And so just keep that in mind, because we don't know what actually they do. So let's talk for a second about ALS and FTD. So these are two fatal neurodegenerative diseases in which there are very specific types of neurons that are lost. Why the very selective types of neurons are lost is not clear, even though we very much appreciate that these two diseases exist on what we consider a spectrum. And that has been demonstrated based on both pathology, immunopathology, looking at the different types of protein accumulations that might occur in the affected neurons in these two diseases. There's one in particular that's in common, and we'll talk about that after, called TDP43. And also there are genetic reasons why we think that these two diseases are linked. In fact, one of the most common variants in ALS is C9orf72. And in fact, expansions in that gene can actually cause either one of these diseases, or there can be individuals that actually will manifest both syndromes simultaneously, or they will develop FTD first, then ALS, and vice versa. Any one of those combinations is not good. Patients are invariably going to succumb to this disease at one point due to the loss of these neurons. So in ALS, the motor neurons that are most affected are those that are the motor neurons that are located in the motor cortex and the motor strip here, as well as the brainstem and the spinal cord. When you think about motor neurons, these are some of the largest cells in the body. In fact, if you want to wiggle your big toe, the cell body to make that action happen actually lives here about mid-spine. So it's a very long axon of about 1 meter. It's the longest and largest neuron in the body. In FTD, it's a different subset. So glutamatergic pyramidal cells, they have particular properties. People try to attribute related to their calcium binding, but that's not what renders them mostly vulnerable. We still don't understand. But these neurons are located in the upper cortical layers. Models of FTD, if you try to model this in mice, are few and far between, because although we understand some genetic components, modeling cortical dysfunction in the mouse is not trivial. Most of the tests that are out there, you have to really work hard to dig for phenotypes. In ALS, because this is affecting the motor circuitry, that's really easy. Does your mouse walk or not walk? So the motor circuit is actually one of the most conserved circuits in the nervous system between rodents and humans. So it's really easy just looking for gait disturbances. Both diseases have very limited treatment options. In fact, most of the treatment options are really about treating the symptoms and just improving quality of life for patients. There are no disease-modifying therapies for either one of these, except in the case for a particular genetic form of ALS right now that has just been recently approved and is extremely exciting for the field. So both of these diseases have inherited and sporadic forms. The etiology is really not fully understood overall. So familial means that there is an inherited mutation. Usually they're dominant mutations. Very few of these are recessive. Though we also have a problem now is that we appreciate that some of the genes don't have 100% penetrance. So you can have the mutation in a family, but you can still end up with family members who do not manifest the disease. Conversely, you could have identical twins, which meaning their genetic composition is identical, and they will manifest the disease at different time points in the course of their life. And why that is not fully understood, but may involve environmental or lifestyle choices. The other main classification for both of these diseases is sporadic, meaning we don't really know what the cause is. There are de novo genetic mutations that have been identified in certain patients. And some of them are in common with those that are found in the familial forms, like C9orf72. But for the large part, for the sporadic patients, we don't have a unified genetic view. We also now appreciate that allogenic inheritance is possible, meaning that there is more than one gene variant that is contributing to the disease phenotype. And of course, there is suspected environmental factors. And so there's, we'll talk about these a little bit later, but there are lots of different things that have been proposed in the field for both of these. There's been no consensus for either ALS or FTD. One of the very popular ones that has surfaced is around head injury, traumatic brain injury. But there are just as many studies that will support that this is actually leading eventually to ALS or FTD as others that will refute that claim. These diseases are fairly common in the sense that for frontotemporal dementia, it's 15 to 22 people per 100,000. This is actually the second most common form of dementia. This dementia is the first one, of course, is Alzheimer's disease. FTD is the second most common and has very distinctive features from Alzheimer's, with executive function being the primary, executive dysfunction being the primary symptom. In ALS, it's a rare disease. It's considered a rare disease at 4 or 5 people per 100,000. But it's the most common of the rare diseases, and in fact, it is the most common motor neuron disease in the world. And this just gives you, these pies are meant to show you exactly what the relative proportion of caseload have familial linkages versus those that don't. You'll see that, so for both of these, the greatest slice of the pie are these sporadic diseases. So we don't have a way to model sporadic disease really well, except for taking skin or blood samples from patients and actually reprogramming them to be neurons and studying those. But there are challenges with using those in the fact that both of these diseases are diseases of aging. And if you're using induced pluripotent stem cell technology, the first step that you do is you reprogram and you erase all of the epigenetic marks, which means that you have lost all of the aging relative marks. So they are good models to look at what is happening in the context of a human neuron, but maybe don't fully recapitulate the aging component that is a primary feature of these two diseases. So in general, we consider these diseases as being a trifecta of genetic predisposition, environmental exposures, and aging. And so this is a model that was put forward by Amar El-Chalabi and Orla Hardiman, in which we consider that every person is born with a genetic load. That is the deck of cards that you were dealt, the hand of cards that you were dealt, and you can't change your genetic load. And so we're all carrying hundreds of thousands of different variants. Most of them are silent variants and do nothing to functional proteins. But some of them do things to the regulatory elements or are actually affecting the coding sequence. As with time, we accumulate, so with aging, our cells accumulate damage, and most people think about reactive oxygen species as being one of those sort of damaging components that we see with time. So if you think just walking around in the sun every day, for example, and then various other environmental exposures, again, in our diet. Do you smoke or you don't smoke? What's your alcohol intake? So lifestyle choices, these, as well as do you live close to an agricultural community where pesticides are being used on a regular basis? Do you serve in the military? Do you play soccer? So Italian soccer players actually have a very high incidence of ALS, as do NHL players, in Canada, hockey players, boxers, football players, and so those are environmental exposures. At some point, they're in the pre-symptomatic phase, but there's going to be some trigger that now the threshold has been breached and that these, our cells are going to actually start a self-perpetuating degenerative process. And so this is where we think about how this intersection between these three components, how are they contributing to disease? Most of the field studies the genetics, and that's because we don't have a good way to model the sporadic form of the disease other than using the iPS neurons that I mentioned. You can also look in postmortem material from patients, but again, that's postmortem. You can't actually really assess mechanism in real time. So most models are looking at genetics. Aging is sometimes considered as, if you're working with mice, you can work with young mice, you can work with older mice, so sometimes that gets incorporated. An environment actually rarely gets incorporated. And that's in part because we just don't really agree on what are the environmental contributions to either of these diseases. So the important take-home message here at this point is that both of these diseases are fatal. And the only way, and I believe, that we're going to actually figure out effective therapies, disease-modifying therapies, is that we really need to understand a deeper understanding of the biology. So how does this beautiful, healthy neuron, this is a beautiful spinal cord motor neuron from a patient, a control patient, so not an ALS patient, versus this very, very sick and dystrophic sort of looking motor neuron. So these motor neurons are shrunken, their cell bodies are shrunken. You do not see great arborization of the dendrites. One of these things is the axon, but you don't see really these large axonal extensions. Something is wrong with these neurons, and how do we stop this process? And we have to do that early enough because we're actually losing neurons as we age naturally. In these disease processes, their system has a lot of compensation that it withins in the nervous system. So I can't run like I could when I was 20. That's part because my muscles are not so great, but it's also because some of the motor neurons that maybe innervated those muscles are not where they were when I was 20, for example. Okay, so back to stress granules. Why is this something that we care about in the context of disease? And so this is for this particular reason. This is so TDP-43, this is the key player in the field that has brought stress granules into sort of the mechanistic aspect of ALS. So TDP-43 is an RNA binding protein. The structure here, it's got two RNA binding motifs, RM1, RM2. The first one is what dictates the RNA binding. RM2 helps to attribute some specificity, but in general, RM1 guides the protein to bind UG-rich sequences. It also has a glycine-rich domain, also referred to as an intrinsically disordered region or a prion-like domain. This is a region in which the protein is very unstructured and can drive aggregation in many contexts. It also has a nuclear localization sequence that is going to drive this protein into the nucleus more often than not. And that's evident here. You see the brown labeling is TDP-43 in the nucleus of a motor neuron. This is in the spinal cord of an individual. And you see that there is a little bit of cytoplasmic granular labeling, but in general, you have the concentration of this protein in the nucleus. That is where it's supposed to be. That's where it does most of its activity. In the context of ALS or FTD, in the affected neurons, you will find any one of these possible various patterns. And so you can see, in some cases, you see very clearly just some cytoplasmic labeling and you've lost the nuclear labeling. Sometimes you have a very, very large inclusion. The nucleus is here in this example. Or you can have skein-like fibrillar type inclusions of TDP-43 or these smaller, rounder ones. In all of these, except maybe this one, the common theme, though, is that the nuclear pool of TDP-43 has been lost. And this would suggest, perhaps, that TDP-43 nuclear function might be should be of a particular focus. And it is now. But actually, for the first 10 years of the TDP-43 work in the field, most people have studied these larger structures, trying to understand the aggregation, because aggregation has been often associated with promoting cell death. And it's true in many systems, it is absolutely true. These various patterns of TDP-43 are observed actually in 97% of all ALS cases. There are only 2 genetic subtypes of ALS in which we don't see that. Those are the SOD1 and the FUS. And in fact, the SOD1 and FUS are both being disputed by a group right now at the University of Aberdeen. But in general, that is our thinking on that, is that you will find this mislocalization of TDP-43 in 97% of all cases, and about half of the FTD cases. And very interestingly, tau is what is the other protein that is mostly mislocalized in FTD, but it is 100% separated from TDP-43. If you have tau pathology, you never have TDP pathology in that patient, and vice versa. Now, this was the initial finding in 2006, was about TDP-43 inclusions. The genetic evidence for TDP-43 actually came a couple of years later, and it came from a pair of papers, one of which was from Guy Rouleau, which is the reason why I ended up moving to Montreal in the first place, was he was one of the groups who had identified a handful of mutations that were sitting within the glycine-rich region. And so what we see now is that there are probably 40 different mutations attributed to TDP-43. They account for a very small proportion of all cases, so like it's a couple of percent. Some of the mutations, you'll see most of them actually are linked to ALS, although there's a couple that seem to track in the FTD population. But in general, TDP-43 mutations are thought to be more relevant to ALS based on the patients than FTD, which is funny because most of the mouse models don't show an ALS phenotype, which we'll talk about later. So TDP-43 is a very big reason why we study stress granules, and that's because some of these smaller structures look a bit like those stress granules that I showed you in the video earlier. And so that's why people sort of got into looking at stress granules in the context of ALS. It's purely by this idea that these things have a visual similarity to these dynamic structures that have been observed in other cells. So one reason, though, I think to think about why was that leap made is because I told you there are genetics that are causative for ALS and FTD, but there's also environmental components. We don't agree what they are, but they're there. So genetics, so there are now more than 45 maybe different genes for ALS. Several of them are RNA binding proteins at this point. TDP was the first one discovered. Several of them, the proteins that are encoded by these genes actually are RNA binding proteins that will localize to stress granules. And in fact, the mutations in them will often enhance the aggregation or liquid-liquid phase properties of those proteins in an in vitro system. And so it was thought that this might be a link. The environment, I talked to you a little bit about that. There are lots of things that are suspected, but in fact, we don't have a consensus of what those environmental factors are. Regardless, if you think about it, stress granules actually are an organelle which actually integrate both intrinsic and extrinsic cues of the environment with intracellular signaling. And so this is actually why I started looking at stress granules, because I thought they were a unique structure in which we could actually maybe start to look at the interplay between genetic predisposition and environmental exposures in ALS. So what is the normal cycle for stress granules? In the video, you saw that normally the proteins are very diffuse. And so I've just listed a couple of the key RNA binding proteins that most people will study. So G3BP1 is a particular focus, and it's paralog G3BP2. When there's an external stress, you're going to have that phosphorylation event on EIF2 alpha that is now going to trigger some assembly or condensation, if you will, of these components. And it's thought that this is happening through a liquid-liquid phase separation process in which you will have several small puncta that will eventually accumulate into larger puncta. This is all probably driven by biophysical processes, and I'm sure you'll hear more about that in a little while. And then when the stress is abated, these things actually dissolve again. The key protein that most people consider the linchpin of stress granule assembly is actually this one, G3BP1. And so it has RNA binding recognition motifs, and it has an N2F or NTF2L domain. This is a dimerization domain, but also a protein interaction domain. It mediates the interaction with a number of different components that help to sort of stick the various RNA binding proteins together, while the RNA binding motifs actually bind various RNAs. And so it's a multivalent sort of interaction between RNAs and proteins that drive these stress granule assemblies. A recent model from Paul Taylor proposes that there are 3 intrinsically disordered regions within the protein that actually that can open, and this is driven by a phosphorylation event. And when they open the structure, now you the RNA binding motif is now available to start binding to various RNAs. That being said, G3BP1 binds a lot of RNAs when it's not even in a stress granule. So I'm not sure how to parse those 2 models together. Through the various proteomics efforts that I mentioned earlier, it is now considered that G3BP1 and G3BP2 are the core stress granule, the key node in the stress granule proteome network. And so this is, I believe, 34 particular components that were identified across multiple studies to be common to stress granules, regardless of cell type or cell stress stimuli. And so you see how this one sits right in the nexus of them all, and this has become an important factor. In fact, you can knock out any of these different components and you're still going to form stress granules, except if you knock out G3BP1 and 2, then you will block a cell's ability to form stress granules. So it's critically important to the assembly of these structures. So this is the normal cycle that I explained. So they form and they dissolve. In the context of ALS, there are now 2 models that have been proposed. The first one is a stress granule dependent model. And so this idea that these structures, these stress granules, when they form, that they transform into the aggregates that are observed in the postmortem material of ALS and FTD patients. There's this thought to be through a hardening of the phase separation properties, so a hardening of the structure. And that this is the reason why neurons are dying. And so, and you can find, I'll go through some of the evidence to support that. Alternatively, there's a stress granule independent mechanism, which is that maybe this key part of the integrated stress response is not happening at all. And that this is part of the reason why motor neurons might be selectively vulnerable and why pyramidal neurons might be selectively vulnerable in these diseases, that we just don't get this process. And so now you've basically, if you think about the cell stress response maybe as a 3-legged stool, you've just taken out one of the legs, and so the neuron is hampered. So that is one of the models as well. And we'll go through some of that data as well. Okay. So the stress granule dependence, so I'll just remind this figure is that this is all premised on the idea that these stress granules transform into these various cytoplasmic types of TDP-43. So the evidence for that. The first piece of evidence actually came from a paper from Ben Wolozin, in which he actually labeled postmortem material of patients and showed that, or claimed rather, that TDP-43, cytoplasmic TDP-43 was co-labeling with TO1, which is one of a common stress granule marker that is usually detected in most stress scenarios. This, the idea that the patterns were the same, and this was shown in a couple of different patients, really drove the field into thinking that maybe stress granules were sort of these, to share Aaron Gittler's favorite term, crucibles of the pathology. It was further driven by another group. So this is from Bob Bowser out of the Barrow, in which he showed that another protein called caprin-1, which is a key interactor of G3BP1, can actually form sort of these cytoplasmic granular things within the motor neurons of patients, and you can see them more clearly here. So these are interesting suggestions, but they could be epiphenomena. We don't actually know that they're linked to the stress granules, but this figure was the key one that drove the field in this direction. This figure has never been reproduced by another group. So localization of TDP-43 mutants to stress granules and cytoplasmic aggregates. I mentioned previously that there are various mutations in many of the RNA binding proteins. We're going to focus on the ones for TDP-43 that actually seem to drive TDP-43 to the stress granules, or they localize to the stress granules at least. And so we see here in cells made to overexpress various mutations, you see that normally you have a nice nuclear localization of TDP43, so the mutations themselves are not disrupting this inherent localization of TDP43. But when you stress the cells, now you see that the TDP43 seems to localize to these structures. So does the wild type, by the way, but very, very weakly. It's actually one of the worst markers of stress granules, but it will go there. And you can actually trick the system if you change the fixative conditions that you could visualize more of those cytoplasmic puncta. In other experiments from other groups, it was looking at the recovery. And so these do the stress granules naturally dissolve, as is what I showed you in that video. Some groups claim that these mutations actually drive the persistence of the stress granules, at least in vitro and cultured cells. Another experiment here, this one coming from Paul Taylor, they've actually used an optogenetics approach in which they drove G3BP1 to spontaneously make aggregates, cytoplasmic aggregates or cytoplasmic puncta. And so they can do that, I can't see here very well, but they can do that by turning on the blue light, and you'll see G3BP1 localize into these cytoplasmic puncta. And through a series of co-localizations, they actually try to establish a kinetics that TDP43 will very quickly, soon after, recruit to these structures, as will a couple of other things that are related potentially to ALS, and that related in the sense that they are also found in postmortem material labeling inclusions in the neurons of patients in both ALS and FTD. One of the key experiments of this paper was the suggestion that the generation of these optogranules that are at the base formed from G3BP1 overexpression actually contribute to a poor survival of the cells following a stress exposure. And so this was part of the key data that drove the field on the idea that these stress granules, where TDP43 has been recruited, are perhaps not pro-survival, and in fact, they should be inhibited as a potential therapeutic choice. This then drove the field into a series of different experiments using small molecule screens to identify drugs that would actively prevent this aggregation. So using high content imaging, there's various libraries have been screened to identify small molecules that will prevent TDP43 puncta formation in the cells and or stress granules. Usually they're together. In both cases, this is actually relying on systems that are overexpressing G3BP1 as a marker or TDP43. Several of these screens that have identified various compounds that will reduce the cytoplasmic localization of TDP43 and seem to be beneficial for cell survival. And it's thought because it's preventing these persistent stress granules from forming. This has actually been the foundation for many companies, including Phase Therapeutics and DuPont Therapeutics, have developed what they call DuPont, they're called CMODs, or condensate modulators, in which they're actually actively trying to interrupt this stress granule seeding of TDP43 aggregation. This system, though, relies on the overexpression of a marker, G3BP1. And so G3BP1 is that core node of stress granule, of the stress granule network, and the stoichiometry of that network is actually really important. Because if you overexpress G3BP1 in a regular cell type, more often than not, without even stressing the cell, you're going to see cytoplasmic aggregation of G3BP1. And it looks liquid-like. If you do photo bleaching, a FRAP experiment, you will see the recovery of the puncta with good kinetics that look like stress granules, but in fact, they're probably just G3BP1 assemblies. So let's consider now the other model, the idea of stress granule independent pathway. So again, thinking about this figure, in addition to all of those cytoplasmic things which draw our attention, right? This is what the pathologists actually focused on. This is how TDP43 was identified, was because we are distracted by big shiny things. And they were just, these very large aggregates was what really caught the attention. But if you look carefully, is that most of these images also show that the nuclear pool of TDP43 is gone. And so this could be considered a loss of function pathway or an independent of stress granules. So what is the evidence for that? And so I'll present, this is a very basic experiment in which HeLa cells are stressed with sodium arsenite. And you see that you can generate stress granules in the cytoplasm as shown by these two different markers, G3BP1 and TO1. And you also see TDP43 will localize to the stress granules. But one of the things that I hope you'll appreciate is that the nuclear pool is still there. And one of the sequelae of events that has been attributed in the field is that TDP43 must first exit the nucleus and then localize to the stress granules. That's not what is happening here. What is happening here is probably a relocalization or reorganization of the cytosolic pool of TDP-43, and the nuclear pool has been completely maintained. And we see that in multiple different cell systems. What about in neurons? And so here's an example of primary motor neurons that were cultured from a mouse that is expressing a single copy of a mutation that is identified in a number of different ALS patients called the M337V mutation. So these neurons have been exposed to sodium arsenite for an hour. And so that's kind of an interesting point, is that everybody talks about neurons as being exquisitely vulnerable. In fact, you have to expose them to arsenite for twice as long as a HeLa cell in order to make them form these structures. So actually, I would argue that they're actually the most resistant cells. They are terminally differentiated. They have every best interest to make sure they survive the exposure. And so they're going to withhold for as long as possible. That's how I interpret that. So we see here in these controlled, the non-transgenic littermate neurons, you see that you have this cytoplasmic signal. I believe this is TL1, and you start to see the stress granule formation. And you'll see that it's also happening on the M337V, but it seems to be less efficient. So you're not seeing as many of the structures. There are much fewer of them. I hope you appreciate the difference between this image in a HeLa cell and this image in a neuron, because this will become relevant to us a little bit later. The stress granules don't look the same in neurons. You do not have these very round punctal structures that you find in most cultured cell experiments. Instead, what you have are much more amorphous sort of cytoplasmic accumulations of the marker. Something to think about for later. Another piece of evidence came from this experiment. This came from Don Cleveland's lab, in which they took SHSY5Y cells. These are a neuroblastoma cell line. They have neuronal-like properties, but fundamentally they are a cancer cell, but they have neuronal-like properties. You can differentiate them and coax them to extend neurites and start to express more neuronal-specific or synaptic-type genes. They genome edited these cells into, they genome edited the endogenous TDP-43 allele to include 2 mutations that effectively eliminate the efficiency of that nuclear localization sequence, and they tagged it with clover. And so in this cell, cell is expressing physiological levels of TDP-43. And you see after exposure with arsenite, you start to see the cytoplasmic accumulations of TDP-43, as well as UPAP2L, which was also tagged. The endogenous locus was also tagged. And you see that these are markers. It's thought to be these are the stress granules. And if you look here in the merged image, you'll see that the green dots and the red dots are not the same. They are actually independent of each other. They are interfacing with each other, but there is not a mixing of the TDP-43 signal with the UPAP2L. So these TDP-43 cytoplasmic puncta are actually independent of sodium arsenite-induced stress granules, which is in direct contrast to what has been proposed by many other groups. These are cell systems. I appreciate that. So what actually happens in patients, and I think this is one of the most strongest pieces of data. So an obligate component of stress granules is actually the polyadenylated RNA that got kicked off of the ribosome in the first place. And so if you were to take patient spinal cord or brain tissues and you just look for where is the polyadenylated RNA relative to TDP-43? In this case, it's a phosphorylated form of TDP-43 that is only present in the pathology, in the disease cases. You see that you have cytoplasmic accumulations of TDP-43. These look a lot like the brown things that I showed you earlier, but there is no signal for the RNA here. So there is no RNA in these inclusions. And lastly, what does G3BP1 expression look like in patient neurons? And so here we have an example of the orbital frontal cortex of ALS FTD patients, and we see in green the nuclear TDP-43 signal. And here's 2 neurons that do not have nuclear TDP-43. And the red dots are individual G3BP1 mRNA. So this is using a base scope approach. And we see that there is fewer transcripts in the neurons that show TDP-43 pathology. And this has been independently validated using a single cell proteomics approach in which G3BP1 was amongst the hits that was actually selectively decreased in the TDP-43 neurons, the TDP-43 mislocalized neurons. And I'll just park this here for a minute. This is a cell experiment that we did many years ago, showing that when you knock down TDP-43 in HeLa cells that are exposed to arsenite, we actually miss this secondary sort of aggregation that should normally happen in the stress granule assembly. We get the initial nucleation, but we do not see that secondary condensation event. And we actually attributed this to a decrease of G3BP1, and it's selective. G3BP2 is completely normal in these cells. The last little bit here is a very recent experiment coming from Peter Todd's group, in which they actually inhibited stress granules using a virally encoded peptide called NSP3. And they showed that in Drosophila flies that are overexpressing TDP43, that inhibition of the stress granules using NSP3 wild type or its control mutant, neither one of them really enhanced the toxicity that was inherent to this massive TDP43 overexpression. And they showed that with a survival assay as well as in the fly eye. You see very clearly actually that NSP3, so inhibition of the stress granules actually worsens the fly eye phenotype. And so suggesting here that actually stress granules are mitigating the neurodegeneration that is inherent in these models. They show it also in a C9 or 72 fly. Perfect. So just 2 seconds left. So this concept of inhibition of stress granules has been pushed forward into the clinic. And in fact, very sad for ALS patients. In fact, earlier this year, we had 2 trials from 2 different companies actually fail. They did not meet their primary endpoints. Both of these are eIF2B activators, which effectively would prevent stress granule assembly. And so both of these drugs actually demonstrate very effective engagement at eIF2B, but there was absolutely no clinical benefit to these patients. Their neurofilament levels, which are an indication of axonal injury or neuronal injury, was completely unchanged in the placebo group versus the treated group. So this was a very big disappointment, but it was a very clear outcome. So the summary of the evidence is that in the stress granule dependent models, there's a reliance on overexpression systems. A lot of what I showed you was in vitro or using cell culture models. And there's a presumption that the visualized structures are actually stress granules, whereas in the stress granule independent, a lot of it was using physiological expression and supported by additional patient tissues and data. And so how to test the models in vivo is what we're going to talk about next. How do we actually really parse that out? And I'll pause there. Thank you. Happy to take questions.
Thank you very much, Christine, for that wonderful overview of the field. I think we have time for some questions. So,
very nice talk. So, can G3BP1 and 2 can complement each other's functions, and depending on what kind of stress?
Yes, G3BP1 and 2 can functionally replace for each other in most contexts, but not all. Is there a question in there?
Yeah, and is there any neuron-specific expression of G3BP1 versus 2?
Oh, great question. Actually, G3BP2 is more expressed in neurons than 1. However, G3BP1 knockout mice actually are embryonic lethals, and the primary phenotype is that they're missing neurons. And it's assumed that G3BP2 levels are unchanged in those mice, but it actually has not been tested. But at face value, there is no neuronal specific expression of either one of them.
Has G3BP ever been seen in the patient inclusions?
No, and I've looked really hard. And so in part, though, it might be a technical problem, is we have used several different antibodies, and we've actually never been able to visualize G3BP1 positive stress granules in tissues, not in mouse tissues or in human tissues. And we think it's an antibody, a limitation of the various antibodies that are available, because we can label with G3BP2 or other markers, but not 1. So no. We've also looked extensively whether there are mutations in G3BP1 in ALS patient genetic databases, and the answer to that is there are some variants, but I wouldn't necessarily say that they were causative. Yes? Oh, yeah, sorry. Just waiting for you to get the mic. Yes.
So yeah, we had a similar observation that we could not see G3BP1 in mouse tissue for some reason, but in rats, we could see them.
Oh, okay.
Yeah, but anyway, so my question was more for TDP43. Do you think that, so what you see is that TDP still stays in the nucleus, a fraction of it, and some of it is outside, right?
Yes.
So is it that those proteins never went to nucleus? That can we differentiate between this, that they did go and then come back, or they are
so it's a great question. Yeah, so the question really is it that when you see the cytoplasmic TDP43 accumulations, is it because the protein left the nucleus, or is it the newly synthesized protein that never got delivered to the nucleus in the first place? And so there's been no direct experiment to test that. I know that Don Cleveland is very interested in that question. We have data, which I'll show you in the next part of the talk, in which we think that it's actually the nuclear pool leaving. But I can't infer mechanism necessarily because they are descriptive, it's observations. But yeah, we don't actually know mechanism.
Transport of TDP-43 has been like, people have given some idea of impotence and so on, but it still seems to be there is some bit of diffusion, right? Which also.
It's definitely diffusion. So TDP-43 was originally described to have a nuclear export sequence, but it was in silico predicted. 3 different papers published actually tested it, and it's actually not functional. And in fact, TDP-43 is 43 kilodaltons, and so is right at the diffusion limit for the nuclear pore. And so there are groups like Jeff Rothstein at Hopkins who really, and Alyssa Coyne, are really pushing forward the idea that it's a defect in the nuclear pore complex that allows for TDP-43 to leak out of the nucleus. And they have good evidence to support that.
Hi.
Hi.
I was just wondering, is there any differentiation between stress granule dependent or independent pathologies with regard to specific type of ALS? Like we see TDP-43 inclusions that are not present in FUS or SOD1. So within the TDP-43 inclusion types between different kind of ALSs, could there be any differentiation in that way?
Not sure I understand. Can you try again?
Yeah, so we are talking about stress granule independent or dependent pathologies with regard to ALS. So just wondering, could it be specific to specific type of ALS?
So I'm going to show you some data in a G93A mouse, which is the SOD1 mutation. That will show you that there are beautiful stress granules in that mouse, and there is no TDP-43 pathology in those mice. Most people say there's no pathology. There's one group that claims there is, but yeah, I'll show you that in a bit.
Hi.
Hi.
Nice talk. Whether the TDP-43 binding to stress granule, does it affect the recycling of the stress granules? Like
Is it affected the cycling?
recycling means
No.
after recovery? No.
No, so TDP-43 knockdown, the cells don't, the assembly is impaired, but the disassembly happens on time. There's no change. Once they finally form, like those little tiny ones, the disassembly is perfectly fine.
So you mentioned a study where you mentioned that inhibition of stress granules worsens TDP-43 toxicity. Inhibition of stress granules worsens TDP-43 toxicity.
The inhibition of stress granules linked to TDP-43 toxicity?
Yeah, it enhances the TDP-43 toxicity.
Yes, in the fly experiment
Yes,
from
yes,
Peter
in
Todd,
both of
and they also have an IPS neuron experiment as well that would suggest that as well.
So what we are saying is that TDP-43 variants, pathogenic variants, would lead to aberrant stress granule formation.
I'm not saying that. I don't say that. There are lots of groups that propose that TDP-43 variants will contribute, that will disturb stress granule dynamics, but those are all using overexpression. If you have physiological expression of a TDP-43 variant, then you have, there's a defect in the assembly, yes.
So they are forming persistent stress granules, some of the mutations.
So the persistent stress granules comes from the overexpression studies. So a physiological expression of a TDP-43 mutation disrupts the assembly. Disassembly is fine,
Okay.
but it's the assembly.
Mm-hmm.
Whereas overexpression, you get persistent stress granules, so stress granules that do not disassemble as they should.
In that case, what could be the possible mechanism of this one, ma'am, where inhibition of stress granules is worsening the TDP-43 toxicity?
What is the mechanism of how worsening stress granules is enhancing TDP-43 toxicity?
Where inhibiting the stress granule formation is inhibiting the,
Oh, inhibiting the toxicity?
yeah. No, ma'am.
Yeah, so I don't know the answer to that. So that's the Matt Disney experiment where they did the small molecule screen. And so they inhibited stress granule formation, which resulted in an inhibition of TDP-43 aggregation, and the neurons were saved. As well as Paul Taylor's optogranule experiment. That might just be that they limited the aggregation. I don't know the answer to that.
I'll go there.
It's a good point though. Like I don't know why, but it's overexpression. But in my experience, I once made a mouse that overexpressed GFP. Those mice were so screwed up because they were expressing like 25 times of GFP, just GFP. And so you overwhelm, protein synthesis machinery, protein folding machinery, chaperone machinery. I don't know the mechanism of what was wrong, but those neurons in those mice were It was not a good model. It was a fail.
So what we are observing by overexpressing in the cells may not be exactly what's happening in the patients.
100%, I agree with that statement.
Okay.
Yeah.
Okay, thank you, Mom.
Interesting. I'm trying to put together the pieces now. So you're saying that TDP-43 basically is not in the nucleus, goes in the cytoplasm, and it's at the interface with the stress granules, but they're sort of independent mechanisms. So what do you think is going on there? Is TDP-43 necessary to, well, actually the depletion of TDP-43 creates misprocessed RNAs that are handled in the stress granules, and that is basically the connection, or?
So I'm going to show you data about how I think TDP-43 is regulating stress granules at the end of the second talk.
All right, so in the interest of time, I think we can move on. And Christine, you can start with this next part, if that's okay.
I have way too many slides, and so I will go fairly quickly through some or probably skip a few, and I apologize in advance. So how do we test this idea in vivo? So a lot of the stuff I showed you about the stress granule, which pathway is this TDP-43 pathology, is it stress granule dependent or independent? It's all been based on cell-based experiments or postmortem material. And so how do we test the model in vivo? And so it'd be a fun fact for you to know that 98% of the literature that studies stress granules has probably used a U2OS cell or a HeLa cell or a hex cell. A couple percentage have used neurons, but all of those are cells that are cultured on two-dimensional plastic plates. They are not in vivo. There's only one other study I'm aware of where they've really looked in vivo. So the environmental factors, I told you at the beginning, I'm really interested in trying to figure out the interplay between genetic predisposition and genetic predisposition and environmental factors. And so these are examples of some of the ones that have been reported as potentially associated with ALS. Smoking is universally associated with everything bad for you, so I don't really consider that one so much. Now, some of these others are sort of interesting. I moonlight on a committee for the Canadian military. Don't ask me how I got into that, but I did. And I got really interested in this idea of the military service and the fact that ALS actually has a much higher rate of incidence in veterans than in those that have not served. And it's especially true in the US military, where the data actually is very robust. And in fact, the US Department of Defense is one of the largest funders of ALS research in the world at $40 million a year. We hope still on Wednesday as well. But also in Canada, where the data is not so robust, but you definitely see that there is an increase of ALS in the military service. And part of my visit to these military labs in Canada was the realization that military service, actually, this ALS incidence is common to individuals that serve in theater or not. So they're on the battlefield or they're not. And it's probably because it's thought to be maybe related to the idea that there's exertional heat stress. Service members are put through incredible training preparedness, including extreme heat and cold training workshops. There's also, so for individuals, for example, that are doing chemical or biological weapon decontamination, they have to wear full body hazmat suits of a completely impermeable material, which raises their body temperature by several degrees. And so there's a whole literature actually trying to understand what are the consequences on the physiology of the service members wearing those suits. Individuals with head injury have been linked with ALS. Excessive exercise, I mentioned about all of the professional athletes, and I talked about it in the context of head injury, but also excessive exercise. There's a lot of ALS patients are in extreme fit of their life. They're marathon runners, they're champions sport, they're champions in their sport. And so this idea of excessive exercise. And then we also, for a long time, have had suspicions about viral infections. And so, but we don't know which ones. I'm not even gonna go there, which ones, because we've all been exposed to viruses a bajillion times. We've all had a cold, so we've had the rhinovirus probably 50 times in our lifetime already. One of the things that I kind of found very interesting after talking with these military scientists was the idea that the commonality here might be heat. And so if we wanted to model an environmental factor that maybe might be relevant in disease, I don't know, maybe we could use heat. It's also the one that our vets would allow me to do. And so we could do, I can't inject the mice with nasty chemicals. The vets do not like that, but they would allow me to put the mice in an incubator, or the spa as we call it, and heat them up for 20 minutes at 44 degrees. This is the temperature that you would normally use to heat shock yeast in a flask. And in fact, you can also heat shock an anesthetized mouse. You need to anesthetize them, otherwise the mice will have febrile seizures. So if we do that, do we see an engagement of the integrated stress response in the spinal cords of these mice? and the brains? And the answer is we absolutely do. If we look for EIF2 alpha phosphorylation, we see that we're seeing it very clearly within these spinal motor neurons. So we're seeing that hyperthermia, this 20-minute acute exposure is activating the ISR. Do we see stress granules? And so this is where I'm going to just quickly remind you about the morphology of stress granules. And this is what I showed you before in HeLa cells, these beautiful cytoplasmic puncta. But I also showed you this in the neurons. They're kind of amorphous. We also have this in a different scenario. We publish this as well. You see these cytoplasmic accumulations or partitioning of the polyadenylated mRNA in the cytoplasm. But they are not the round droplets that we are accustomed to seeing. So can we visualize stress granules in these hyperthermic treated mice using various markers? And so one of the markers we chose was HUR. It's commonly found in most of the RNA stress granule paradigms. And here in this case, you actually do see really beautiful droplets of HUR. But you can look with several other markers, including G3P2, AF3N, and TR, and we don't necessarily see the same beautiful cytoplasmic puncta that are nice and large. They're usually of a certain size. You see really, really small ones. And this brings me back to sort of like this idea that stress granule composition is highly variable between cell types. And even if you try to co-label any of these markers, you're not going to see very much co-localization of these markers in a mouse spinal cord. However, if you looked in a HeLa cell or a U2S cell, you'll see almost 100% overlap of the markers. So that is something that we don't understand of why that's different in vivo, but it definitely is. But it also raises the question, so which one of these markers do I follow? I don't know. And also I remember a friend of mine, Sami Barmada, always says, antibodies suck. Your antibodies are only as good as how you've characterized them. And most of the characterization of antibodies are not done in mouse spinal cord tissues. And I didn't have access to the knockout mice of these various ones to know for sure that that signal is specific. So the other obligate component of stress granules, of course, is the polyadenylated mRNA that is kicked off of those ribosomes. And so if we track that, you can see very clearly that the cytoplasmic pool partitions in the hyperthermic animals. And so this became the marker that we've used extensively in our studies to try to understand how our TDP-43 pathology is it stress granule dependent or independent. So here's an example of just blowing up that same image. And you see that in this 20-minute heat exposure, more than 80% of the neurons in the mouse spinal cord are actually demonstrating what we call stress granules based on this poly A labeling. And we show that also using a MATLAB script that we wrote to look at edge detection because I wanted to see another method. And it's definitely, we see stress granules quite well. So what happens in the ALS mouse model? Or what happens in ALS mouse models? So there are two mouse models. The workhorse of ALS for many years has been this mutation in SOD1, superoxide dismutase 1. This G93A mutation is not particularly common in the patients, but it's one of the most commonly used mouse models. In fact, it is the #1 preclinical mouse model to test drugs in before they go to clinical trial. This mouse does not have TDP-43 inclusions, nor does it have nuclear clearance. It has a very bona fide motor neuron degeneration that is late onset, progressive. The mice will die of what clearly looks like an ALS phenotype. Now, TDP-43 mice, there are easily at least 24 different models in the literature. None of them generate a motor neuron phenotype that looks like this one. None of them either generate TDP-43 pathology. They don't have nuclear clearance and they don't show cytoplasmic aggregates. There's one mouse that does show nuclear clearance in aggregates, and this is an artificial mutation in that nuclear localization sequence, and that mouse is expressing 10-fold of the TDP-43 levels over control. That mouse is a great mouse for certain questions, but not for this one. So this mouse has no TDP-43 pathology, but this isn't a bona fide ALS mutation. So, oops, there we go. So this mouse, one important part to know about this particular mouse is that TDP-43 is very well known for splicing, and I think you heard that from Aaron Gittler earlier in the week. And so it can, in a loss of function scenario, you can actually get incorporation of particular sequences that are previously unannotated, these cryptic exons. And you see that here in the TDP knockdown. These mice do not show cryptic exon splicing of this particular mouse gene target. Neither does it show skiptics, the idea that overexpression is going to drive the exclusion of particular exons, and we don't see that either in these mice. So that's really interesting. This suggests that the expression of TDP-43 has been maintained. The physiological levels is maintained. In fact, in other data I'm not showing you, we actually show that the mouse allele is actually downregulated in these mice. So this is a knock-in of a human TDP-43 mutation into the ROSA 26 locus, and the autoregulatory components are maintained. And so as human TDP-43 is expressed, the mouse comes down, so you end up with a constant steady state level of total TDP-43. And you see that there's no disturbance in the splicing patterns in these mice, which is very important. So we're not overexpressing and we're not underexpressing. So what does stress granule formation look like in these mice? We're using this acute model. Young mice, we see that, in fact, the cytoplasmic polyadenylated RNA is not partitionating in these mice, whereas in the G93A mouse model, it is working just fine. And we can quantify that up beautifully in that we see that the G93A mice are just as efficient at making stress granules as non-transgenic glutamates from either line, whereas the mutant, the line carrying a TDP43 mutation is not showing stress granules at all by two different measures. We know that the IF2 alpha phosphorylation is intact. We don't really see any significant differences in the engagement of the ISR, so whatever is happening is not upstream. In data that I'm just going to summarize here, we looked to trying to understand what could be the possible cause for this impaired stress granule assembly. And in fact, what we found was that in these TDP43 mice, we have a decrease in various RNA binding proteins that are usually required for stress granule formation, including G3BP1. We also saw that the cytoplasmic pool of mRNA seems to be decreased. And you can kind of catch, these are shot with the same settings, and you see that that cytoplasmic pool is not nearly as intense here as in the control animals. We also saw that the RNA chaperone EIF4A1, which is known to actively inhibit stress granule formation, seemed to be increased in these mice. Now, I don't know which one of these mechanisms or which combination of these three are actually driving that. It could be all of them. It could be just one of them. This is based on a drawing by Roy Parker. This group is the idea that what we have in these mice is we have less free RNA than typical. We have less RNA binding proteins that are typically found in stress granules than normal, and we have an increase of the RNA chaperone. Any combination of these, we just do not cross the percolation threshold to form stress granules in this model. That's how we published this story, because we couldn't work past that for the mechanism of what was really going on here. What we know is that they're impaired. So what about TDP43? That's the whole question, is what happens to TDP43? And so in non-transgenic mice that are exposed to the hyperthermia, every now and again, we would find these neurons that seem to show a complete nuclear clearance of TDP43. You see diffuse cytoplasmic labeling of TDP43. These are in the spinal cords of these mice. But we don't see TDP43 inclusions. And in fact, what we saw is that this happens way more often in this mouse model that's expressing a TDP43 mutation. And we really don't see it so much in the G93A as quantified here. This fits very well with what other groups have published, that there's no TDP43 pathology in these mice. And this here we have in this mutant mouse, we actually are starting to see TDP43 nuclear clearance, but no inclusions. This mouse, though, at baseline has perfectly normal TDP43, as you see here. So this is actually the first time that a mouse model of expressing a TDP43 mutation actually demonstrates the nuclear clearance of TDP43 and it's stress responsive, which is interesting. So to summarize this part, so these mice, we see that we do not have stress granules and we do not have TDP43 inclusions in these mice. We see nuclear clearance, but no aggregation. And in the G93A, we have stress granules, but no TDP43 inclusions. And so this data taken together is very inconsistent with the idea that TDP43 pathology is a stress granule dependent process. So there are limitations to this work. First of all, this was an acute terminal stress because it was 44 degrees, the mice were anesthetized. We cannot actually evaluate the persistence of the stress granule. There's no recovery in this experiment. It is a stress and an immediate sac to look at stress granule formation. So we can't evaluate stress granule persistence and the link to TDP inclusions. It doesn't address the most important question in the field, which is, are the stress granules neuroprotective or are they neurotoxic? Because that's what's going to drive what type of therapy we actually work on, what kind of therapy we develop for patients. So to get at that, we revisited this idea of the different stresses. And so I think for those of you that exercise on the regular, you don't just do it one and done, you do it recurrently, right? You do it repeatedly. And so the idea of recurrent exposures, and we have repeated stress granule assembly and disassembly cycles in cells, and what the cells eventually sort of like, okay, I've had enough. Like, they get tired and they can't execute the pathway anymore. The other component that needs to be considered is aging. Aging is actually the #1 risk factor of ALS. It is the one thing that everybody agrees on. It's the #1 risk factor of all neurodegenerative diseases, but let's just stay on ALS and FTD. Should also say FTD. So can we incorporate these two things in our studies? And so in the acute model, there was a piece of data that I held back until now, which was that we noticed that in the young mice, you see these beautiful partitioning of the cytoplasmic polyRNAs, beautiful stress granules. And if you look in mice that are much older, so 18 months is quite old for a mouse in a colony. The natural life of a mouse in a vivarium is about 24 months. So at 18 months, we see clearly that we're not seeing that partitioning. And in fact, less than half of the neurons are actually going to make stress granules if you age the mouse out, which means that there's a natural decline in the neuronal ability to participate in this stress response mechanism, which is interesting and could be relevant to what we're looking at. So can we develop a new model to look at this recurrent exposures and incorporate stress? And so one of the things that we did here is we lowered the temperature a little bit. We kept the mice awake, an important part. They're restrained, but they are awake. And we can give them multiple exposures of hyperthermia. And so in order to first demonstrate that that worked, we had to lower the temperature. We wanted to lower the temperature so we avoid febrile seizures. We see, can we form stress granules after 60 minutes at 40 degrees? And we definitely do. You can see them very nicely here, both by Poly-A and HUR. And then if you just give a 15-minute rest, 15 minutes of putting the mouse back in its cage, not in the incubator, you actually can see complete dissolution of the stress granules, suggesting that the assembly and the disassembly is working perfectly. So we do that now. We're going to do that twice. We're going to do 2 consecutive cycles. We're going to do that twice a week, and we're going to do it for 4 weeks with the idea of trying to mimic the recurrent stress. And one of the things that I hope you'll appreciate is that we didn't see a lot. We saw TR puncta here, but we didn't see stress granules so well. So this is when we look at the animals here after the 4 weeks. So they've gone through the multiple cycles, and then we sacked the mice. But that makes sense because the stress granule, we allowed them to recover, and so we didn't really expect to see that. So we didn't see stress granules, but we saw TR puncta, which was confusing initially until we thought a little bit more about the fact that we see them regardless of the age of the mice. We see them all the time. Sometimes we'll even see them in animals that are not even exposed to the stress. And so TR is often found in these cytoplasmic granules, and we can count that up. And you see that you can see that that happens quite a bit in the non-transgenic. It also happens in the TDP43 mice, and maybe it's maybe exacerbated by the presence of the mutation. This is preliminary data still, but there's no partitioning of the cytoplasmic mRNA in these mice. We're not seeing stress granules, but we clearly see TR puncta, which raises the question of when you're looking at TR puncta, what are those? I don't know. The important question, though, is if you have those TR puncta, if they, for example, are the persistent stress granules that people have reported in the in vitro experiments, what happens to motor neuron survival? And the answer is nothing. There is no change in the motor neuron counts in these mice when we look out several months after the heat exposure. And we can look in young mice. We can look at older mice that are exposed for a month. We can look at mice that are older and then allowed to recover. We see there's absolutely no significant difference in the motor neuron counts, suggesting that the TR cytoplasmic puncta have nothing to do with whether or not a motor neuron is going to live or die in this scenario. Well, what about TDP43? So just to remind you that what we saw in the acute model was that about 30% of the neurons were showing the cytoplasmic TDP43 signal, but no inclusions. And so here's what we see. We see 2 different patterns. We see either that complete nuclear clearance, like I showed you before, or we see the preservation of the nuclear signal as well as cytoplasmic puncta, and we can have that in both. Interestingly, this is a phenomenon that you see the nuclear clearance, you see it in the early stage in the early mice at 4 months, not so often in the older mice, but now we see the puncta are happening much more often in the older mice, whereas we see no puncta at the 4-month stage, consistent with what we initially published. So we see that we can actually reproduce TDP43 pathology in this mouse when we actually integrate the genetic predisposition of this mutation, aging, and this environmental stress exposure. It also, in red here, you'll also see that the TR puncta that are labeled are actually completely independent of TDP43. So those TR puncta that I showed you in the previous slide, they have nothing to do with TDP43. They are completely separate entities. What about motor neurons? And so I want to focus here is in these transgenic mice is that we see in 12-month-old mice exposed to 1 month of stress and assayed immediately. We see a selective loss of the alpha motor neurons, which are the ones that are at risk in the disease, whereas there is no change in the gamma motor neurons. These are the ones that are more related to postural muscles, whereas these are the ones that are enabling you to walk, for example. And you see that with recovery, as you allow the animal to age a little further, is that you actually see that this becomes even worse. And so we move from 30 to 50%. And so we have selective stress-induced motor neuron loss in this mouse. This is a mouse model that at baseline shows absolutely no motor neuron loss in the presence of this stress at either age. So to summarize, this is the integration of these 3 components. And we see that we have nuclear TDP43 clearance occurs first. And then we seem to see that there's cytoplasmic TDP43 puncta that occur later, but the motor neuron loss seems to be directly correlating with the amount of nuclear clearance of TDP43 that we have. And so we can see the TDP43 nuclear clearance by heat stress. The puncta manifests with aging. So this is the first model that incorporates aging. These puncta of TDP43 are independent of TR, and these TR puncta have nothing to do with motor neuron loss. But TDP43, alterations in TDP43, whether that nuclear clearance and or the cytoplasmic puncta seems to be correlating with selective alpha motor neuron loss. So coming back to this model, we think we've settled part of it. We've put data forward now supporting a stress granule independent mechanism. Stress granules are protective, and these TDP43 inclusions that are observed are absolutely independent of stress granules, at least in this model. So can restoration of stress granules maybe be something that is a therapeutic target? And I'm out of time, and I will just maybe skip to the end about how this works. Is that I mentioned to you before about TDP43 regulating G3BP1. It doesn't regulate G3BP2. We can also see that in an expression of a mutation that we're lost G3BP1. I'm going to skip through that. And just show you here that TDP43 actually can bind in the 3' UTR G3BP1 at a conserved element. It's conserved all the way to C. elegans. And in fact, we even validated in C. elegans because we have a worm lab next door using TDP knockouts. It is definitely conserved. And we can also show that TDP43 is actually stabilizing this transcript, and it's doing this through the 16 base pair sequence that we can delete and actually show that we can restore the stability, which is shown here. I have a second story about how TDP43 also regulates the cryptic splicing of TDP43, which I don't have time. I'm out of time. And so I do. Oh, the clock is still saying, oh, I'm so sorry. I have 15. Okay. Okay, thank you. Okay, so I'll still go quickly, but maybe less quickly. Okay, so this sequence that's sitting in the 3' UTR, it's actually a UG-rich sequence that we spent quite some time trying to characterize. And actually what we found is that there's actually a microRNA sitting right in the middle of that sequence. It's called miR329. It's a neuronally enriched microRNA that is conserved it's highly conserved between rodents and humans at least. And if we use a mimic of that, we show that we can drive the loss of G3BP1 mRNA without touching TDP43. So we really show that this microRNA has action on G3BP1 RNA. Using EMSA's experiments, we published this already, that TDP43 binds this region of G3BP1 that encodes this UG-rich sequence. We also show now that the microRNA binds in experiments I'm not showing because we're working on the stoichiometry, but it does look like there's a competition between the microRNA and TDP43, with the microRNA having a tighter binding than TDP43. So it suggests competitive binding. This experiment is a cell-based experiment where this is the only experiment you're going to see from me where I've overexpressed TDP43 because I do not like to do that. So we overexpress TDP43 or an RNA binding deficient mutant. And what we found is that when we treated with the mimic, is that this particular mutant actually cannot counteract the mimic for this miR329, whereas overexpression of the wild type overpowers the mimic, and so you get increased levels of the transcript compared to this RNA binding deficient mutant. What happens in the context of cell stress? And so if these are now N2A cells that have been transfected with the mimic, and we look at what is the percentage of cells that form stress granules. So if we overexpress miR329, this is going to result in a decrease in G3BP1 protein, which I'm not showing you, but that is true. And you will see that we have a decrease in the number of cells that are able to form granules in response to arsenite. And this actually translates to an exacerbation of arsenite-induced cell stress or cell death. So again, sort of feeding the idea that stress granules are protective. So we think here we have a working model that TDP43 normally binds in that 3' UTR and stabilizes it. And it's doing that by sterically inhibiting the binding of a microRNA. And so this, we think, presents a therapeutic opportunity that if we could interrupt the way that microRNA binds that sequence, we could now stabilize G3BP1, even when TDP43 is not in the nucleus in the first place. And I know somebody here is going to say, but microRNA degradation happens in the cytoplasm, and I'm talking about nuclear TDP43, and in data not from us, but from others, showing that TDP43 binds the G3BP1 transcript in the nucleus and stays bound to transport it out to the cytoplasm, and it has to stay bound. And so we don't understand, so we're a little fuzzy on that mechanism, but the microRNA inhibition is clearly restoring G3BP1 expression. There's something that we don't quite fully understand here. So we're looking at the idea, can we normalize G3BP1 expression as a potential therapeutic target? And we're using that with John Watts, who's an ASO person. So we're developing antisense oligonucleotides that can compete with, in the context of cells that are stably knocking down TDP43. We have one here in which we see that this ASO can sterically seems to be sterically inhibiting TDP43, the microRNA, and we're setting a restoration of G3BP1. These ASOs are currently being delivered into mice in which we're knocking down TDP43 using other ASOs. So we're excited to see whether or not we can actually prove in vivo that we could rescue G3BP1 function. And then, of course, the ideal experiment would be then to take those back into the hyperthermia model and actually show that when we reinforce the stress granule pathway, then we can actually preserve those neurons that we saw as lost in the previous model. So that's still data to come. So I've handed you a beautiful story, gift after the bow, with in vivo modeling. It settles the mechanism of stress granule independent TDP43 inclusions. I show you that TDP43 is stabilizing G3BP1. There's a potential therapeutic strategy. Done. You heard from Aaron Gittler earlier this week how TDP43 probably does a lot of different things. And so I told you about this part of RNA stability and stress granules, but the number one thing that people think about at TDP43 has to do with splicing. And in the course of our studies, one of this type of splicing, pardon me, is this idea of cryptic splicing. The idea that you get incorporation of previously unannotated exons, and that can result either in the generation of a normal protein, but maybe the protein is lost due to loss of the transcript due to nonsense-mediated decay, or you could maybe even get an in-frame insertion, for example, which is something that I'm going to tell you a little bit now. So in the course of our studies, studying this set of TDP43 binding sites in the 3' UTR, we saw that there was this other set of peaks that were sitting in an intron. And for the longest time, I ignored them. I wanted nothing to do with them. And then there was another group that kept saying, you know, I think maybe you need to look at that. And so this is from Michael Ward and Pietro Fratta, in which they had identified that in their TDP knockdown neurons that there was this cryptic exon that seemed to keep popping up in neurons that had TDP43 knockdown. We took a little bit of a deeper look at this, and we saw indeed there is this in-frame insertion. This is a 30 nucleotide, so 10 amino acids exon. It's got canonical 3' and 5, pardon me, that's labeled wrong, 5' and 3' splice sites. And immediately downstream is that we see a pair of TDP43 binding sites. And we thought, okay, we better look at it, because it was also, if you dig into the data set that's available from Pietro Fratta's group here, in which they looked at TDP knockdown, and they reported the UNC13A cryptic exon, and also Aaron Gittler also reported this. We also saw that G3BP1 is sitting right there, is one of these dots. So what is this cryptic exon real? So the first thing for me, I thought it was junk. So we approached Alyssa Coyne at Hopkins, who has a collection of about 130 different IPS motor neuron lines. And we asked her, do you see the cryptic exon in these lines? And in fact, in lines in which she has already observed TDP43 cryptic splicing of 13 other genes, we see that there's definitely an increase of the cryptic exon. This is 111 different lines here, whereas in lines in which she did not see the TDP43 splicing signature, we don't see it. We don't see it in controls. We also see it in the C9orf72, which are patients always have TDP43 pathology, and so that tracks quite well. We don't see it in the SOD1 cases, which tracks well with what I showed you in the mice, that is TDP43 independent. And there were only 3 lines with TDP43 mutations, and so we can't make a conclusion because it's statistically underpowered, but we think there's also relevant there. If we look now in the New York Genome data set, so this is looking at 2,000 cases. We look in the motor cortex and frontal cortex. We see clearly that there are patients that have an ALS or an ALS diagnosis and TDP43 pathology have a higher rate of inclusion of this cryptic exon in the motor cortex, and a trend in the frontal cortex. We don't quite hit significance there, but we're awfully close. So we think it's real. And so why, how does it work? This caused us a lot of grief. And so when we realized serendipitously, a postdoc was doing TDP43 knockdown in iPS-derived neurons. And we had sort of a dose-dependent knockdown of TDP43, depending on how much of the TDP43 lowering ASO she was using. And we saw, originally she was doing the experiments here at 5 micromolar, so we were seeing a knockdown, but I never saw the G3BP1 total RNA go down, which we always saw in all the other experiments. If we doubled the dose, though, now we start to see the G3BP1 total RNA go down a little bit, not a lot, but a bit. It's 20%, which is actually the same amount in which we see a decrease in the mouse model that we're using. However, what we saw is that even with the low dose of TDP43 ASO, we actually have the cryptic exon is actually popping up. It's getting incorporated in G3BP1. And so it looks like it's a concentration-dependent increase. So we see the cryptic exon inclusion before we see the decrease of the RNA. And just as a positive control with Stathman2, which is another cryptic exon that's reported, we see very clearly the Stathman2 cryptic being incorporated. And at the protein level, we can verify, for example, that the cryptic, we really see that TDP43 induced loss of G3.1 only at the high dose of TDP43 ASO and not at the low dose. So we have cryptic exon inclusion, but we do not have a disruption in the G3.1 protein pool, which means that we have a pool of the protein that has an insertion. This insertion is 10 amino acids in the N-terminal NTF2L domain. This domain, actually, the site is exactly where caprin-1 is predicted to bind. And in fact, the insertion sort of displaces how the caprin-1 peptide would insert. And so just to remind you where caprin-1 sits in the core network. And so if we do IPs, we see very clearly that if you express the cryptic exon in cells that are lacking the endogenous G3BP1 and 2, we see that we've completely lost caprin-1 binding. And so we would predict then that stress granules should be disrupted. And so first we did a proteomics experiment using Apex labeling, and we see that we have a very large decrease in the binding of various core stress granule components in dark red and other components in the lighter red. And you see there's a very significant shift to the right. The cryptic exon is on a gross level disrupting the stress granule network. What happens to stress granule assembly? So cells here that are, again, that they are a double knockout for G3P1 and 2. And if we overexpress uniquely either the wild type or the cryptic exon included form, we see that the wild type can rescue arsenite-induced stress granules, whereas the cryptic exon does not. And if we repeat the experiment now using parental cells, we actually see that in cells that are expressing the cryptic, it actually functions as a dominant negative and prevents stress granule formation in these cells in which it's expressed. So one of the difficult things with when we found the cryptic and why I was initially super not excited about it was that we could not see it in most cell lines. And so the two cell lines that we often use for the in vitro experiments, U2OS and HeLa cells, we see beautifully TDP43 knockdown. And in both of these cells, you'll also see the G3BP1 destabilization effect of that. But we can never visualize the cryptic. Like we never got anything, we never got a signal, but we could see UNC13A, which is a very well-described cryptic exon. And so we realized after a while that maybe we should be thinking more about it. Is there something neuronally specific? And so there are a handful of neuronally enriched splicing factors that we thought maybe we should just do a little sequence gazing and have a look. And we actually stumbled on this SRM4, which actually has multiple binding sites within proximity of that cryptic exon, including one that sits right in the middle of the bipartite TDP43 sequence. So now if we create a mini gene and we overexpress SRM4 in U2OS cells, in which you will not normally have this event occurring, we can actually see if we can get inclusion of the cryptic peptide. And if we look now for the endogenous, so again, using U2OS cells in which we're stably expressing an inducible form of SRM4, we can now generate the cryptic peptide, the cryptic transcript, pardon me, in these cells that normally it was not present at all. And just for control, you see that TP43 levels are completely unaffected by SRM. So is this a second therapeutic opportunity? So the idea of using a steric ASO to prevent the cryptic splicing from being included, I'm not sure, I don't know. We have a hard time to integrate this with the previous model, but there are now papers that are reporting that you can have TDP43 splicing changes observed without having a gross relocalization of TDP43 into the cytoplasm. This was shown by Jenna Gregory at Aberdeen using aptamers, and she shows actually that the nuclear pool of TDP43 can redistribute within the nucleus, and that is sufficient to generate Stathman2 splicing. And so we wonder if that could also be the case here. We consider then that maybe that this cryptic G3BP1 is maybe fine-tuning G3BP1 function, and we talk most often about G3BP1 function in the context of stress granules, but it's been reported in a couple of other scenarios as well. And so most of the actually, fun fact is that most of those stress granule proteome experiments using the Apex labeling approach, for example, the proteomes don't change that much between the stressed and the unstressed cells. They're always sort of interacting with all their partners anyways. It's just whether or not they coalesce into these larger structures. And so we're wondering whether or not this is a way to sort of fine-tune G3.1 function. So I don't know the answer to that yet. So we've shown here dual regulation. TDP can regulate G3.1 either by splicing or RNA stability. And so in the normal physiology, it actively represses this cryptic exon. It actively stabilizes the transcript. In the context of pathology, in a dose-dependent manner, you can actually see that it vacates, the TDP43 vacates the spot, allowing SRM4 to enhance the inclusion of this exon. Or in the case of a much larger nuclear clearance, you see that we actually have microRNA-mediated decay of the transcript. Either way, all roads lead to Rome. Both of these mechanisms, in fact, impair stress granule formation, impair the ability of G3P1 to make these structures that we think are inherently important to neuronal survival. And so if it's compromised, we think that this is a contributor to neuronal vulnerability and loss in the disease. So the final slide, we've shown you in vivo modeling that favors stress granule independent model for TDP-43 pathology. We firmly believe that stress granules are neuroprotective and should be promoted at all costs. G3P1 is regulated by TDP-43 by two very distinct mechanisms. And so is this a potential therapeutic strategy? Maybe. I think it's complicated with this idea of two different mechanisms. And I think we have a lot of work to do to figure out how to translate this from fundamental biology to an actual therapeutic strategy. Last slide is thank you to my team. Esmita and Hannah are two postdocs that have led the cryptic exon story. Alicia Dubinsky is just going to be leaving my lab in a few months. Is the lead on all of the in vivo stress granule work. She's an absolute powerhouse. And then Mariam and Adrienne are a PhD student and a master's student who have really driven the microRNA story with one working in the mice, one working in human. And there are nuanced differences between mouse and human for G3P1 that I didn't cover here. And lots of collaborators that I'm incredibly grateful for sharing reagents and time with me, and of course, funding. Happy to take questions.
Thank you very much, Christine. I think we can take a few questions.
Go ahead. I'm going to take the first one up top here.
Hi. Yeah. Okay. So with regard to the transgenic TDP-43 M337 mice, is it expression only in motor neurons or other neuronal cells also have this expression?
It's universally, it's using the genomic promoter. It's all expressed everywhere.
How do you think that stress granules protect from heat stress?
Great question. I don't know the how of how. I mean, it's been proposed that there's sequestration of apoptotic signaling. That's one mechanism that comes to mind, but I honestly am not sure. But it would be an important next step to try to parse this out. We're also trying to develop another stress to see if it's common to a different stress paradigm. Yes. Yes. How long does it take? So with the ASOs, 24 hours. Yeah, we transfect in the ASOs, and 24 hours later, we clearly see that we've preserved G3P1 transcripts. That's in progress right now. Yeah, what I showed you was the cell screening we were trying to pick the best ASO. But because we've discovered some nuances between mouse and human, we've decided, screw it, we're going to put them all in the mice and see which one is going to work. So I don't have the data for that yet.
So given what I took away is that the different genes that are associated with ALS, they seem almost to work quite differently. Given that you have such a strong loss of splicing function for TDP-43, does that help you unify these other pathways rather than focusing on stress granules as the critical target of TDP-43? There must be very broad splicing changes.
There's, yeah, hundreds of splicing changes, 100%. It's not, yeah, so I should not have, don't want to mislead that G3P1 splicing and regulation is the be all and end all. I'm very focused on stress granules for this talk. But TDP-43 regulates 1,000 different transcripts, either through differential expression or splicing. There's no particular theme around them. So UNC13A is a synaptic protein. Stathmin2 is a microtubule stabilizing protein.
G3P1 just broad splicing defects. Maybe you don't need to put a gene name on it. It's just
it could be exactly. It could be, and so that is the reason why some folks are actually just going after looking at putting TDP-43 back in the nucleus, just reinforce that and solve all of it, rather than doing these sort of first layer, target specific events. One of the complicated things that I didn't highlight at all is, the splicing, what genes TDP-43 regulates for splicing and expression in mouse and human is wildly different. It's not fully conserved. And so, is the fact that none of the TDP-43 models really faithfully recapitulate an ALS phenotype linked to that? maybe. Like Stathman2, which is a very highly motor neuron enriched gene, that cryptic event doesn't occur in mice. But a humanized mouse model, they actually were able to show that you could get motor neuron loss. So maybe that's part of it.